Hello class, this is Mrs. Smith, and today's lesson will be about the scientific method. At the end of this lesson, you should be able to know the steps of the scientific method and how and when to use it. The steps are shown on this slide: the problem, the background information, hypothesis, experiment and its various parts, and the conclusion. The trickiest part of following the steps are knowing what to put where. The problem is always going to be a question that you want to answer. The background section comes next, and that includes the research that you're doing on your certain problem. You want to find out if someone has already done an experiment and what they've learned about this particular question. The hypothesis is your proposed answer to the question. That means it's what you think will happen if you impose the certain restrictions, or what you think the answer to the question is. The experiment will require you to do some careful observing, then make sense of what you saw, and the conclusion is putting it all together to write a synopsis of what you learned about the question. You want a final answer. Designing an experiment is trickier than it might seem. Remember, you're trying to answer a question. So when you ask the question, think about what kinds of materials you'll need to investigate the answer. The independent variable is the part of the experiment that you change on purpose. The dependent variable is the part that you're tracking or keeping data on to see if it changes as you manipulate the independent variable. That means that the dependent variable is going to be the data that you collect. The independent variable is the part that you're changing on purpose to see if it has an effect on the dependent variable. There are different types of controls that you want to understand before you impose controls on your experiment. Sometimes in an experiment there's a control tube. That means that you're going to leave one tube alone with just the original chemical inside of it and use that as a basis for comparison. Some experiments include a control group. That means that there's a group of test subjects or a group of tubes or beakers that you don't do anything to, and that remains under normal conditions. In the example on the screen, if you have a fish tank full of sick fish, you would leave 2 of the fish in the tank with the original water while you take the other fish out and try some different cures to see if those things work on the fish. And experimental control are ones that you've most often dealt with in biology class so far. So an experimental control means that the condition is going to be the same for all groups, the control group and the experimental group. So for example, all plants in the experiment are kept at the same temperature. That is going to eliminate temperature as a cause for any change that you might see in the data that you're gathering. After you design the experiment, it's time to get some data. There are 2 different types of data: qualitative data and quantitative data. And the differences are just found in their name. Quantitative data means quantity. So you're going to have something that's measurable with a number. Qualitative data is more the quality of what you're seeing. Make sure that you're consistent and regular in your measurements and observations. You don't want to start something in meters and then change to centimeters. So always use the same unit, measure on a regular day, etc. And a chart will help you stay organized, so a lot of times we'll have data tables that you'll need to fill in. After you have your data, you want to analyze it. The graph on the left shows you that these 2 variables are linked, so temperature and reaction rate are related. They have a direct relationship, meaning that if there's an increase in temperature, this data is showing that there's also an increase in the rate of the reaction. The graph on the right-hand side has the data points very scattered, and that means that these 2 pieces of data are not linked. Temperature and size do not affect one another. There are a couple other types of relationships that we'll talk about more in class, including an inverse relationship, an exponential growth relationship, and an exponential decay relationship. This experiment shows an example of how you could use the scientific method in your everyday life. If you want the batteries to last longer on your Wii remote, you need to do some research first. Then you will form a hypothesis. So here's a sample hypothesis. If I turn the Wii remote off every time I use it, the battery will last 30% longer. The hypothesis does not need to be phrased in the form of an if-then statement, but sometimes that's helpful. To further this example, the independent variable would be turning the remote off. That's the condition that you're going to change on purpose. The dependent variable, again, is the part that you're going to measure. So you will measure how long the battery lasts. The controls that you want to impose, the experimental controls, are that you will use the same user and the same amount of time being played for the control group and the experimental group. Once you have the experiment designed, you will want to gather data. Your baseline data with your Wii Remote left on in between playing sessions, that's going to serve as your control group. And then, again, according to your hypothesis, you're going to turn the Wii Remote off in between playing sessions, and then you'll note how long the battery lasts in both cases.